See a strand of seagrass. Hardly changed depth at all since we've started the dive.
Have we changed up since the start of the dive? Yeah.
Not too much.
Yeah, we're now at 1207. Yeah, 07 plus altitude, so 1209.
Another thorny head.
It's clearly getting rockier. There's just a lot of sediment draped over everything. Would your guess be that these are encrusted?
Yeah, definitely see some of those bubbly textures and black colors. Hard to tell.
Bridge nav, can we add another 50-meter step on that? Thank you.
Steve or Greg, what is the name of the very large purple and silver urchin? Is it a formosoma or something different out here?
I think it's Asthenosoma, A-T-H-E-N-O-S-O-M-A. Yeah, it'd be good to get one of these dives, but no rush today.
Was there another request coming to you guys in the back row? Thought I heard something vaguely.
Yeah, right at the moment. We were talking about the big urchins.
Oh, okay.
That Greg would like one eventually, but doesn't have to be today.
Okay.
A threesome.
Before the pilot throws it in your lab, it says, hey, do this.
What? A lot of sea urchin, sea cucumbers.
Yeah.
You could call this Benthodeides trail. Wonder what it is they like here.
Our oxygen saturation steadily declining.
That's a good point. So is this just like a giant lava flow? Or
Yeah, I think since
sheet
this is,
flow of some sort?
yeah, volcanic origin, northeast bank. Flows, what Or maybe
Asteroid,
it's crust.
White asteroid, yeah.
thorny head.
Bridge nav, another 50-meter step, same bearing. Yep. Thanks. Do you guys have the option to see the DP screen back there? So it would be DP2? I don't think you do.
I don't think so.
I don't see it. The main.
Looks like a sponge, maybe a Faria sponge. Anemone.
It's interesting ledges. I
This,
mean,
the ledges, the bare parts of the ledges have most of the life.
yeah. Looks like good ferromanganese crust too. The Argus view really shows these larger ledge features.
It's pretty ledge-like in the Herc view too. Oh, there's definitely loose rocks.
Yeah, here's some.
Assume they've tumbled.
How far have we
Sure.
gone along the transect, Aaron?
We're really close to the end. 8 meters to go.
Cool.
Do you want us to hold at the end? Are you thinking rocks?
Might be interesting to get a deeper one. But let's see, we're only at about 1164.
If we see something that's easy to pick up at the end of the transect, let's try that.
Okay.
Otherwise, if
Well, I would say
if
we're...
it's just like this, we would.
Okay. We are end of transect.
Okay.
There's a ship move in, so it'll keep moving, so keep your eye out. And call if there's something you want to try for.
Yep. 6.
Is that sea urchin again? Oh.
There's a nice rock.
That is a nice rock.
Is the fly trap an enemy on there?
Yep.
Yeah, I'm happy to, if there's any of these at the base of this boulder or...
Pick up that whole thing.
Bridge nav, hold position. It's a rock.
yeah, we can look at the biology on this outcrop.
Oh, a cesta maybe? A tanner crab?
Sorry, Bob, you're not on SPL. Couldn't quite hear you.
Yeah, Steve's confirming cesta clams.
Yeah, we're gonna blow right by me here. So,
Yeah. So
well, I can't.
Can't stop here. That's okay.
We need to move back if we're gonna.
I'll keep step moves now smaller so we can stop faster.
No, it's okay.
Well, yeah, so we're gonna blow right past here. I can't.
I know,
do
Bob. I know.
anything.
Thank you. I got it. We got it.
Well, I'm just saying you can try and catch it by moving back, but
Yeah, so I see how the edge there was really polished.
Shiny.
So the currents are really flowing up and over that boulder picture. Probably why there's so much life there, catching stuff on the passing currents.
You got the end of the transect down.
No, I didn't.
I'll bring it back up.
Thank you.
That was the problem. We were trying to write and look at that rock at the same time.
Missed the time, but
Bridge nav. 20-meter step 312.
cool. Thank you so much.
Yep.
Well, it's interesting to see on the slope map that we're going through this mottled region, and it's
Yeah.
pretty much telling us it's a mix of sediment and rock.
Yeah, I do think that is like just the general rugosity showing up. Rugosity, the ruggedness of the seafloor. Oh, sorry.
Sound like a Star Wars character.
I like that. I like that word.
That'd be a good name for a dog.
Is this, have we caught up to
Similar
ourselves?
sea urchins.
Yeah.
Is this somewhere we could
Could we zoom
take
in
a look?
here?
Can we stop?
Bridge and nav, hold position.
See some of the
Still,
megafauna
we're
here.
still gonna blow by.
Thank you.
Greg, do you want us to collect this sea urchin? He left the room, so I'm not sure if he's listening or...
They might be trying to
Are you sitting down?
We're getting there.
What,
Take
do
a
you have
look.
something to tell
Yeah.
us?
I know.
What are you going to tell us?
If you're down, yeah.
Okay.
Can that go in with the sea cucumber?
Yeah.
Thank you.
It'd be really cool if we could grab one of these rocks off the ledge,
Let's
but
get a rock also.
bet they won't break.
Yeah, Bob, can we
Those
get a...
cucumbers are
yeah,
nasty to touch, though. We've got the purple.
What? Anemones are?
The cucumbers,
Zoom.
they're
Yes,
very
please.
spiky.
Really?
Iris down a little bit.
They're not soft and fluffy.
all right, good captures.
Wait, these ones? Is this a cucumber and an anemone?
I'm sorry, it's a sea urchin.
Okay.
Did I say
Okay.
sea?
We good on that?
Yeah.
Ready
That's
to zoom
great.
out?
Thank you.
It's an urchin,
Okay.
but they are spiky.
Is something gonna squeeze out of it when he grabs it?
I'm gonna try, and this is extra challenge because it's not working exactly right.
It'll be a kind of squishy, Bob, so don't worry about breaking it. Should be okay.
Roger. Do you,
Soft, right? Ish.
Greg, do you have special gloves to handle anemones in the lab?
I think I'll get Kyla to handle it.
Special gloves called grad student, is that?
We'll just use tongs.
Fancy. Okay, got it. I want one of these rocks.
An urchin or a rock?
I'll take the, I want one of the rocks. Like, love that one.
is hard doing it this way.
See how soft the test is. It's compressing when he picks it up.
Yeah, he's squishy.
It's like
Looks
a
like
balloon.
a balloon.
Right? Yeah. Jinx. There he was, just minding his own business.
Wow, he's really squishy.
There we
Oh,
go.
there he goes. He's out of here.
Oh, oh.
He's also almost neutrally buoyant.
It doesn't look like that's what should happen.
Oh.
He outcast.
Just blew a hole. A hole
Yeah,
just
it's-
opened up.
Sorry.
Where's he going?
It's going in a box.
Yeah, 6 white on the
Tool tray out?
forward box.
Oh, whoa,
Oh,
whoa.
shoot. Oh, shoot. I don't know what to do.
There's nothing you can do.
Keep it.
Sorry.
Yeah, that's the problem.
With the floaty
Yeah,
stuff.
you warned us they were fluffy. Okay. Good to know.
He's in the gauges. If you want to...
I'm not fishing around back there.
Okay,
no.
fair enough. Sorry.
He's in the
But-
gauges.
Tool tray in.
Okay.
Maybe he'll be there when we get to the surface.
I don't think so.
Oh, gosh.
We couldn't even keep the rock from the front porch yesterday.
Yeah, we can work on that. If we do another one of those.
Get a rock here?
Uh,
Oh, I forgot to mark my sample.
gotta move up a little bit.
What's the sample number for the urchin?
NA124, 068 for the sea urchin.
Correct.
Cucumber doesn't know how lucky it is.
That's a Moby Dick story. Or to the jaws back.
His friends are never going to believe him.
Which rock?
Wait,
Kira?
there's one over
show
there.
Me,
On the,
show me what you're interested in. And
that one's actually got a lot of. Can we zoom in?
this guy? Right here?
No. Sorry.
Well, it's got a dead sponge on it, I guess. Right?
Yeah.
Like that rock?
Yeah.
Okay. Zoom out just a little bit more.
Oops. Zoom out.
Was the sea cucumber sample 067?
Yep.
Yeah, I guess it's got a pretty good. What's all that sediment stuff on the front?
Those are hydroids and things on
biology.
living on it. Yes, biology.
Darn biology always
Oh,
getting
you
in the
don't.
way of
You
my
don't
rocks.
want that. Oh, okay. If you're just looking
Well,
for
no,
rocks,
no,
we got.
We like it.
Lisa likes it, so.
What's that all that other stuff on
Can you
it?
can you pick up that rock? We'd
Is
like
that too
it.
big? It would be a good one. Wait, where are you going?
I'm moving over so I can get to it.
Oh, okay. Yeah, let's definitely get a picture of context. That one has
Yeah.
more macro than others in the area.
That would be selectively choosing our rocks.
Good.
non-representative. I do a little bit worried the sponge skeleton will just topple right off it, but we'll see.
All right, can we zoom in? Give it a second to clear.
Let the dust clear.
We'll just see if it will budge first. Yeah, it's budgeable.
Snowing. Will it fit in our insert?
We have to
That
look
is
and
the
see.
question. It
So
may not.
it's pretty long.
We only have 15 centimeters.
Yeah. So it's, well, how deep is the box?
Well, it could go in it could go in 5 red forward, the forward box, possibly.
Yeah, I think the sponge is probably not going to stay attached, right? That
But
looks like
that's okay. If it falls off, that's okay. In the box.
Yeah.
Tool tray out,
Okay.
coming out.
Do you have any guess of what type of rock that is, Kira?
Yeah, it's a very manganese encrusted rock. Is my educated guess.
Do we have good pictures?
Nope, we're losing brittles.
Get some grabs.
Yeah, I've got some grabs.
Thank
Got
you.
it. Almost looks like a scallop, but maybe that's a nice, I think it's a scallop, a cesta. Serpulids, lots of brittle stars, maybe bryozoan on there. Some other cool things.
A lot of brittle stars on
Zoom, zoom out.
Rock 1, woo.
How was the sponge skeleton last night, Greg? Were there a lot of things there?
Turn it a little.
Too big. Oh.
Nope.
Oh.
Hey,
There we go.
Cool.
nice one.
Tool tray in.
So we're going to call that rock 1. Yes?
Yep.
I'm using the same target for that one, so no change in position.
Okay.
Okay, so R1 should be with sample ID NA124-069.
Thank you.
All right, ready to truck?
Yeah.
I'm going to do a
Thank you.
slight heading change to 300.
Okay.
Bridge nav.
I wonder if it would ever be interesting to take 2 rocks in the same location, one with a
Can we
lot
get
of
a
mega and one
20
without,
meter
and see if
step
the
bearing
microcommunities
300?
are different or similar.
What did she just say? Missed it.
Sorry. What'd you say, Aaron?
I was just putting in a ship move.
Okay.
So we're going 300 for 20 meters, or until you say stop.
So that sponge again looks like Berea, glass sponge.
Another fly trap an enemy.
Uh-oh.
What's that? What's that?
High flyer.
Giving a hold.
What is that?
What is it? Fish, is it part of a fishing line?
Looks like possibly. I don't know.
There's weights on that line?
Can you zoom in? Doesn't really look like fishing gear to me.
No, looks like somebody else's
Science
science
gear.
gear.
There are these tilting current meters that
That's probably what it is.
look a little bit like that.
Should probably mark
It's
that.
anchored
Yeah,
to
I'm gonna drop a
something.
target.
Somebody's going to recognize that. Maybe somebody on the internet.
It's like the type that Chris Roman uses.
Well, that must be the weight there. Yeah, those are, right? Looks
Mesh
like
bag of weight.
lead weights. You could use those.
Is that in a
Yeah, we
mesh?
could. We need those. Are we allowed to pick up things we find?
No. What is that? That's weird.
That's weird.
Like chafing gear.
Is it okay to call in another move? You're not worried?
Yeah,
Okay.
I guess.
Bridge nav, another step, same bearing.
Oh,
Oh, what is that?
Wait,
Wait,
Oh wait,
what
there's
was it?
something at the end of it. It's not a dead whale. What is it?
Yep. What is that?
Is it an old mooring?
Oh, it's somebody's mooring. Yeah.
You can zoom in more.
Just looks like the line is disintegrating. It's an urchin tucked under there.
You know what it might be is a sonobuoy, a Navy sonobuoy. I mean, we're in Navy territory here, right?
Yeah.
Yeah, that's what it is. They drop them out of airplanes to locate submarines.
All right.
All right, I guess we leave it there.
Yeah, they drop a bunch of them.
You know, Apex floats, like Teledyne Apex floats that do profiling. They drop those out of planes too.
Yeah, they drop these out of the P3s and the S3s. That's totally what it is. All right.
Ship's moving. Bridge nav, another step, same bearing, and sorry for letting my mic open.
Did I do something?
Correct, thank you.
guess that's just part of it. Zoom in.
Some garbage.
More
It's like
foil.
tinfoil.
Yeah, that's all part of the sonobuoy, I'm sure. Can we zoom in on this?
Or sponge.
Pretty.
This is Faria and one other thing. Notice how the brittle star is just like the dead part of the sponge, not the live part. Beautiful. I
Okay.
don't think we can take that rock though.
No. Moving on.
Do you see those nice big bubbly textures? This is more
Yeah.
what I was hoping. I mean, those are really large botryoids, but nice pair of manganese crust textures, especially on the
Biggest
cards
fly
with
trap.
nothing
Big
on it.
Look
shrimp.
at
Ooh,
that guy.
that's pretty. Can we zoom in on that beautiful anemone? Or whatever
Hey,
it is.
zoom in.
Cereanthed?
Is Steve here? Does he know what that is?
I don't know, Steve. Do you recognize this? Paralomus underneath. Pretty anemone though. Got a good spot.
That's beautiful.
Okay.
Good?
Yep.
Yep,
Thanks.
good
Carry
captures.
on.
Yeah.
The anemone was? Wow.
Bridge and I have another step, same bearing. It's nice.
Your depth here?
A lot more sponges now.
There's more rocks for them to live on.
Now we have 4 bio boxes left.
Yeah. You probably could put something in with the urchin. I think there'll be lots of rocks up closer to the summit.
Yeah, I was just thinking if it's worth trying for a depth distribution or physical biology diversity.
Do you think these are different than what we just sampled?
Could be.
Right.
Not sure. I mean, I'm not saying here, but if there's any plan to
Yeah, I think
sample at depth intervals.
I think the other watch will have lots of rocks to choose from. Next watch.
Guess that's the stock.
Seems like
So
often that,
we're gonna change watch.
yeah.
Bridge nav, another step, same bearing.
What's fascinating is how each place we go has a different community. You know, and Site 4 on the Patton Escarpment was kind of a similar trek, but completely different fauna. It's especially at the base.
Yeah, we're down to 4.2% oxygen saturation right here.
So these sponges, which I think are Faria, really do well in low oxygen. See them in Costa Rica, and all sorts of places. Almost like a low oxygen specialist.
Zoom in.
That's pretty scenery. There's some really long transparent tentacles. I wonder if there's terabellid inside.
It's a big old shrimp to the right there too.
You can see the really fine botroids too on the surface of the rocks.
Yep. Can we have that rock?
Which rock?
I'm
sort of kidding. That big thing that they're all on.
Oh,
Oh.
okay. Got it. Can I reset your DVL?
Yeah.
Ooh, could you zoom in on the lower pink worm-like creature, please, so we can see what that is? Is that a giant aplicophoran? The Greg is probably not watching.
He's right here.
Look
What's
at
the
it.
pink thing?
That's what I'm wondering.
He says it's a sea cucumber.
It's a cucumber?
Mm-hmm.
Oh. You don't think it's a large aplicophoran? It's got the line down like a meniomorph. It's got the line down it. Should we get it? Could we slurp that pink thing, please?
Zoom out. Hey.
Changing of the guard here.
Will it?
It's big. Yeah.
Should we switch
You ready to?
before or after the slurp?
Do you want us to record this slurp and then?
We can switch.
I'm not a Beats fan.
We're undergoing a bit of a shift change here.
Yeah, Argus is like
video test, looking good.
I don't know if that works, yeah. Pretty far. So science, that's my understanding. We're going to go for that sea cucumber there.
That's correct. We think it could be a strange kind of mollusk.
Oh,
We'll see.
interesting. Roger that. I'm going to set a reposition so that we are able to actually reach it.
Ooh.
Was that me there, Jonathan? Sorry.
I think it was me.
Oh, Raj.
Okay. Jake, it's never bad until you see the sediment move, then
Yeah.
Then you gotta move.
Yeah, let me know if you don't.
That's pretty good.
We used to have the tether short enough that, and we were calm enough that that's kind of the way we operated. It was pretty low to the ground. But
Yeah.
Happy Halloween, everybody. We've just had a shift change. We've got a new crew driving, managing the Hercules and the Argus. And in the back row, the peanut gallery is from Scripps Oceanography. And there's myself, Greg Rouse, Paul Jensen, Caitlin Creamer, and Kyla Pearson. And we'll be here for the next 4 hours till the end of this dive, and we'll be heading up this slope of what's called the northeast bank, and we're on the eastern side of it. We're still pretty much at the bottom of the slope at 1,240 meters, and our goal is to try and get up to the top of the slope, a flattened plateau, because this was once an island.
All right, Jonathan, before we take a slurp, go ahead and take a picture, please. Oh, slightly occluded.
So if it has a shiny surface, it's covered with spicules. Oh, it's going back into the rock. It looks like it's pretty big. I think it is an aplocopheran. So this is a kind of mollusk that doesn't have a shell. It looks more like a worm or a sea cucumber than a mollusk, but it has gills at the back end, and at the front, which I think is lobed up towards us, is where the mouth is, and it has a scraping tool inside that really tells us it's a mollusk. This thing's probably preying on these anemones that are just above.
Oh, interesting.
But it's pretty unusual to see them this size. This is going to be an interesting thing to have up in the lab tonight, hopefully.
Raj, full wide there, please, Jonathan. All right, Jake, go ahead and flush it a little bit here, please.
Roger, flushing.
See if I stabilize or not. Are you 100% right now?
Yes.
You want to back it off to 70?
Sure.
Okay.
What jar is this going into, Science?
One of the coarse ones would be good.
Could we put it in jar number 7, please?
Yes.
All right, Jake, I'll have you do 70% again there, please.
Okay.
The
70%.
sample number.
Reg, stand by.
This sample number will be NA124.070.
Jonathan, go ahead and push in there, please.
070I.
Great. All right, Jake, go ahead.
70%.
Reg. Oh, hello.
Very small.
He's on to us.
Come here. We just want to sample you.
Oh, I think it went in.
Kind of hard to see, but hopefully.
Is it going? Going?
I think you got it. It couldn't have pulled away that quickly.
Traveling through the tube.
Yes.
There it is.
There
Woohoo!
it is. All right.
Awesome.
Oh, wait, Jonathan.
We're off to a good start.
Fantastic.
Definitely aplicofferum.
Perfect. So that's going to be NA124.070 or S7H1842. Slurp number 7.
All right. Anything else around here that you guys want samples of, or are we going to just keep trekking?
Let's keep going. Thank
Roger
you.
that.
Great slurp.
And Rennie, what's going to be our next bearing?
We're going to go 305,
305, Rog.
and I'll let you get a little bit in front before I move.
Roger that. There we go.
You're going to get out in front before I should turn my heading?
Yeah.
Okay.
Well, you can actually go ahead and turn your heading now.
Roger.
I'll just look on DVL.
305?
305.
Yeah, it's only like 10 degrees anyway.
Got myself a bit of a mess there. Jonathan, you can stand by. I'm just trying to get myself a clean.
Sorry about that. Just trying to clear the bottle.
Roger that. Coming up.
Looks like you,
Coming up.
the last watch got some pretty nice sediment cores there.
Oh, yeah. Definitely. Jake, you want to go back to the front porch view there, please?
Yep.
Thank you.
Might take a bit to move, so I'll go ahead and call that in.
Roger that.
Bridge nav. Step 50 meters, bearing 305, speed 0.3. Thank you. See how that is.
Yesterday we had a great dive on a volcano west of here called the San Juan Seamount. And we came up the western side of that, and the last half of the dive we were really in amongst a fantastic sponge and coral extravaganza. Unfortunately, we planned to dive there again, but the wind blew up, so we've come inshore somewhat, and we're now at this place called Northeast Bank, a bit south and to the east of where we were last night. San Juan was once an island, and so was this Northeast Bank. So it's really a flattened platform on top. We're still at several hundred, 500 or more meters below that. We will be lucky if we can get to the top, but our goal is to get up much further up this slope. But we'll be sampling rocks along the way and trying to gather other interesting animals. The goal of this cruise is to document the biology associated with encrusting minerals on various kinds of rocks. So there'll be ferromanganese incrustations, which is likely what we'll see today, but also phosphorites. And these are potentially for mining in the future. So, but it's good to understand what we have associated with those. And our microbiologists along on this cruise are interested in how these crusts could be formed with microbiology involvement. And maybe Paul can tell us a bit about what he expects to find from that front and how he's been sampling to get a handle on how these microbes can be involved in forming these crusts.
Yeah, sure. Hi, everyone. So there's a group of us on this cruise who are microbiologists, and we're interested in, broadly speaking, microbial diversity in the deep ocean, in particular in ocean sediments, and associated with these various mineral-rich substrates. So these are unusual communities, and one thing we're particularly interested in is the possibility that some of these microbes are producing things that we call natural products. These are small molecules that bacteria are really good at making, and many of them turn out to be useful for us as medicines and other useful purposes. And so one idea is that some microbes may be attracted to these mineral-rich surfaces because they use them to help respire, basically. It sounds funny, but they basically breathe rocks. When there isn't a lot of oxygen around, they need to transfer electrons to something, usually that's oxygen. And when there's no oxygen, they may be transferring those electrons to these various metal oxides. So, so we're interested in getting a sense of what sort of microbes are on these surfaces. And then we're going to use molecular techniques to ask what their potential is to produce various types of natural products that may have useful purposes. So in the context of this project, we're calling this biopharmaceutical potential. And there's potential both in the microbes that are living in these sediments and on these surfaces, and we want to explore that potential. And there's also potential in the various invertebrates that we see. So when you look at a reef and you look at the animals that are there, you'll see some that have spines and shells and claws and other obvious structural defenses, and then you'll see others that lack those obvious structural defenses, and they don't have a way to escape predation. They often rely on chemicals to defend them against predation. And so we'll be collecting some sponges and other types of vertebrates, especially certain groups of soft corals, certain octocorals that have a history of making these kinds of molecules and using them as chemical defenses. Things are very different in the deep sea than on a coral reef where we usually work, where predation pressures are much higher, but nonetheless we're interested in asking these same kinds of questions down here and addressing the biopharmaceutical potential of both microbes and some of these invertebrates that we're hoping to collect today.
Never seen that before. It's like I'm going to have to restart this thing. Yeah, not before I have some fun. What?
That reminds me of a spirograph. Remember those?
Yeah, that's not good. Not what you want in navigation software, spirograph. Jake. One demerit, Jake. Spooky. Yeah. All right, back in business.
So to the pilot and subcrew, some of the things we're keeping our eye out for today are we'd like to get a collection of that mushroom coral that we saw yesterday, and so you guys can help us keep your eyes open for that one.
Sure thing.
And also there was a gorgonian and octocoral we saw at the end of yesterday's dive that we hadn't seen yet and haven't collected yet. So I don't know if we're going to get up shallow enough to have a chance to see that animal again, but if we do, that's definitely something we'll be looking to collect.
This is a very cool rock, lots of really smooth formations here. Some of that shiny crust that we're looking for, really cool. Large rock covered with some different biology. One of those sea urchins that we've been seeing.
Curious that is like a statue out here, you know?
Yeah. Is this kind of all by itself? We just kind of stumbled upon this.
Yeah, it's all by itself out here.
Wow.
Yeah, I'll talk about it.
It's got some different creatures on the different sides. Very cool. Yeah, you can see that view really nicely from the Argus cam. It's a big boulder.
Ooh.
Ooh.
Looks like a Pharea on a stalk, but I'm sure it's something